Hello, this is Ramu Anandakrishnan. I'm with Edward Via College of Osteopathic Medicine in Blacksburg, Virginia. I'll be presenting our study where we showed cranial manipulation affects the expression of neuronal systems genes in an animal model of cognitive decline. Dementia, as you're aware, is a growing problem worldwide due to an aging population. There are pharmaceutical treatments, but due to age-related comorbidities, the adverse effects often outweigh the clinical benefits. So there's a need for alternative approaches. Osteopathic cranial manipulative medicine offers such an option. In a previous study, we had showed that OCMM can improve spatial memory in aged rats, which is shown in this picture on the right. In this study, we want to understand why. So we compared the gene expression levels in brain tissue samples from treated animals to those of untreated animals. And what we found was that several genes in the acetylcholine neurotransmission pathway were overexpressed in OCMM treated animals compared to untreated animals. Interestingly, this is the same pathway that is targeted by acetylcholinesterase inhibitors for treating Alzheimer's disease. So I'll now go through some of the details of this study, starting with our overall approach. So for an animal model of cognitive decline, we used aged rats, 18-month-old rats, and we used the Morris water maze to assess spatial memory. So at the end of this behavioral study, we then took tissue samples from the prefrontal cortex of euthanized animals and compared the gene expression levels of the OCMM treated animals to those that were not treated. So as I mentioned in our previous study, we showed that OCMM treated animals, shown by the red line here, outperformed the untreated animals, shown by the blue line, on a Morris water maze assay, where we measured the time that it took the animals to find the escape platform. To why we compared the gene expression levels in treated and untreated animals, and we identified 426 genes that were significantly differentially expressed with a false discovery rate of 0.004. We then looked at the pathways associated with these 426 genes, and what we found was that neuronal systems genes were overrepresented in this set of significantly differentially expressed genes, as shown on the right figure here, which suggests that the OCMM treatment affects the expression of neuronal systems genes. We then looked at the top 5 genes in this set of differentially expressed neuronal system genes, and what we found was that all 5 of them were part of the acetylcholine neurotransmission mechanism, is illustrated below, where the 5 genes are identified. Interestingly, as I mentioned earlier, this is the same pathway that is targeted by the acetylcholinesterase cholinesterase inhibitors used to treat Alzheimer's disease. So this suggests a possible mechanism by which OCMM could affect neuronal activity and thus improve the performance of aged rats. We also did a literature search on the 426 significantly differentially expressed genes and found that 40% have been previously implicated in neurological disorders, which is shown in this figure on the right, where the genes associated with or implicated in dementia are identified in blue, movement disorders in red, and psychiatric disorders in yellow. So this again suggests that OCMM affects the genes that are differentially expressed due to OCMM could affect the symptoms or progression of other neurological diseases as well. So in summary, in this study, we tried to understand why OCMM improved the spatial memory in aged rats, and what we found was that the treatment seems to affect or increase the expression of several genes in the acetylcholine neurotransmission pathway, which is the same pathway affected by Alzheimer's disease drugs. And we also found that many of the significantly differentially expressed genes, 40% of them, were previously implicated in neurological disorders. So these findings provide justification for further investigating OCMM as at least an adjunct treatment for age-related cognitive decline. With that, I'd like to thank VCOM for their generous support, and also Virginia Tech for access to their sequencing center and animal care facilities. And this is a reference to the previous study where we showed that OCMM improves the performance of aged rats in a Morse water maze assay. Thank you.